Hudac. I'm from the United States. And I am so excited to be here and share a little bit about what we're doing actively upstairs right now at this conference. So I wanted to just start to give you a sense of where I come from, because I'm going to talk about EEG in a little bit of a different way. So just to give you a sense of where I've been around the United States, I was born in Mars, and then went to college and started to get really interested in mental health, and then trained in MRI for many, many years. So I was working with kids as young as 4, getting them into the scanner, autistic kids. And I realized not all of our kids can make it through an MRI scan. I think a lot of parents probably can attest. So I ended up switching and moving more into EEG in grad school, working with infants primarily in the hospital, but found myself missing this autism piece. So ended up doing a postdoc more on the genetic side, so working with a multitude of different genetic conditions that are linked to autism and intellectual disability specifically. So I kind of had this autism bias more so than epilepsy, so just to note that as I talk. And I'm now at the University of South Carolina, where we're able to do this work en masse with a team of people. So with generous support from the foundation, we were actually able to bring multiple people. So why I can give a talk while they're upstairs collecting data right now. You've probably, some of you family members might have heard from my coordinator, Liv Mace. She's fantastic. And Dr. Niccolo Pini is our ace in the hole, because he's Italian and is able to translate everything. And I'll kind of share the study as we go along, and you'll see this QR code again. If you're a family member who wasn't convinced to sign up for the study, there's still slots. We'll make it work. So feel free to use the link or find me after the talk. So I want to talk a little bit about why we care about the everyday brain, and there's a lot of important need to be using EEG to understand seizures and sleep, but we spend most of our time really thinking about how the brain works in our kids to really link back to then what we see in our animal models. So I wanted to just kind of give you an overview of kind of our perspective, and I apologize, this is a very common slide for anyone who's heard me talk before. But we really look across this wide range of things, and we know that it's more at the kind of protein and genetic level that our pharmacological interventions are going to work. But when we think about our kids, we're really starting at this top-level diagnosis. And these could be lots of different things. These could be mental health conditions. This could relate to seizures or GI issues. And a lot of these are things that we're actually going to intervene on more on that behavioral level, right? So I don't want us to forget the fact that we have amazing therapies and therapeutics that we can be having our kids do that aren't just the drugs, maybe together with our drugs, right? So kind of understanding that this is a complex problem, and our whole goal is to figure out the best outcome for our kids. So if we think about something like autism, which is where I mentioned I started my career, this is something we diagnose behaviorally. There's no blood tests for autism. It's something we see based on how individuals act. And so what we spend a lot of time doing is thinking through, can we create brain-based indicators that are really specific to autism? So we're kind of coming down in this funnel, and we use a lot of different tools. Like I mentioned, I've done MRI before, but now we're really using EEG as a kind of a nice central point, and our goal is to figure out biomarkers that are really specific to autism and do not measure neurotypical development, and really say, oh, here's how I would stratify between autism and neurotypical development. But what's really exciting, and I'm super thrilled that my career has taken me here, and we're not inventing this. There's lots of giant consortiums, including the EU AIMS here across the European Union, that are developing biomarkers. But a lot of times those biomarkers are not for our kids, in part because of intellectual disability. We're seeing IQ cutoffs because they're trying to start with kids that are more communicative verbally, right? So we have a basis, but it's not in our kids just yet. So what I've been doing is really coming from what we call a genetics first perspective, where we start by trying to understand groups of individuals, and here I'll talk today about GRIN2B more specifically, but using this to understand, are there shared etiologies that link together where maybe we can, in the middle of this, kind of join forces. So using our work as a bridge between that clinical side and the preclinical models as well. So that's what I'm hopefully here to do. I will do my best to talk each of your languages, but I sit firmly in the middle here. So when we think about GRIN2B, this is not something I need to over-explain. We've already had lots of fantastic talks overviewing what we see phenotypically. So we also see hypotonia, motor delay, which we haven't talked about a ton today yet, microcephaly. But some of the work that we've done in talking with parents and looking at behaviors is also to identify some strengths. So this is some work that we did just using a parent-based measure. And here, having an elevated score indicates more social skills problems. And what you can see is related to social motivation. So how interested and engaged our kids are with other people. We actually see normal or neurotypical-like levels in our GRIN2B populations. And that's relative to other gene groups that I work with, as well as autism groups, both with a low IQ and an average IQ. But just because we see these amazing social strengths, but we still struggle with some social boundaries and how to interact with people. And we see poor attention and sensory behaviors as also kind of being a challenge behaviorally. So thinking through then, how are we going from having a GRIN2B disruption and kind of building up? Well, we know that the kind of molecular changes are leading to how our brains are built. And there's some little tiny bit of MRI work. So here's a citation from a paper that found this atypical fold, so how the hippocampus is formed is atypical here, which is a really important brain region for understanding and maintaining memories and learning. So what we try to do in my lab is to say, okay, well, here we're seeing learning and language at these two different levels. Can we use something like biomarkers to truly bridge and understand those two pieces? And where we've started, it's not the only biomarker by any chance, but I'm hopefully going to convince you of today, is that thinking about attention and then also looking at how that's related to online real-time learning is a fantastic way that we can do this and kind of build that bridge with our EEG-based biomarkers. And if I have time, I'll talk a little bit about the work that we're doing cross-species with the Netta Shanks group to look at the fruit fly. So what is our BioGENE study? As I mentioned, it is currently happening upstairs. I think we're on our eighth participant of the weekend. I work with other genes as well, and our website has a little bit more from each gene group. We have a specific GRIN2B page. We will be looking into expanding that because, as you can see, we have lots of families have signed up that aren't just 2B, so we're really, really just getting excited to learn what's different and what's the same in our kids. And we use the same EEG system, and I have lots of videos to show you how easy it is for us. We do this because we know that the glue is not going to work for our kids. So this net goes on within seconds, and depending on how well we did on that first go, we might take maybe a minute or two and adjust some things, but then we are immediately collecting data. So even if you have a kid who doesn't like wearing things on their head, which is a lot of our kids, usually we have toys and things and can help kids re-regulate, and even if we get 2 minutes of data, that's fantastic, right? So we're looking for anywhere between 5 to 20 minutes of data, but whatever we get is worthwhile, which I think is really important to emphasize. And I will say that in the past 3 or 4 years, I think I can only think of maybe 2 or 3 kids who haven't given us a sufficient amount of data to be usable. And that's out of hundreds of kids. So this is really, really exciting. The other reason why we really like EEG is that it's developmentally scalable. So one of the things that is a big problem and a concern for us is the fact that GRINs could look different at different time stages. And we're going to hear a little bit more about mini puberty, and we spent a lot of time talking about puberty in my group. So these are all times in which our brains are changing, we know, and so how our kids are changing differently in these groups, it gets really complicated. So it's great to have a measure that can kind of really scale across those different levels. So I started the BioGENE study when I was in my first institution, and COVID hit, so if you can tell the timing of fall 2021, brand new professor, was excited to jump into my first big time project. COVID delayed things. And we are working with kids that are medically complex. So I did not want to fly 50 families to Alabama. So I packed up my car and started in Seattle and decided I'm going to drive all across the country. And I did. So I drove a little bit of everywhere, and I'll even show you. I converted to kilometers for everyone here today. It was exhausting. My father was very concerned about my car's battery and health, and it made it. And across 3 months, I saw 55 kids and 1 adult in their home. One family preferred to do it in a hotel, so that was fine, but I was able to kind of make this an accessible thing for them, and it was entirely because of these families below here that are leaders of the 2 groups I was working with, Families SCN2A Foundation, and then I think many of you probably know Liz and Phil Ash, who run the GRIN Foundation in the States. And it really was the parents posting on social media and saying, hey, this person showed up in my house, and here she is doing EEGs. And I thought I would just show you a little bit of what this looked like for those who haven't seen. I practiced in my friend's basement for a couple of days just to get fast enough so that I wouldn't take up too much time bringing all the equipment in, because I was just one person doing this. And then this just gives you a snippet of how different each of the kids were. And we're doing it in the kitchen, we're doing it on couches, in standards. This child on the top left, that was at 5 in the morning, which is not my happy time, but that was her happy time. So I was there at 4:15 to set up. We had kids that were able to have lengthy conversations with me, and then kids who didn't. Kids that were fully wheelchair users. We had this child who fell asleep, and that was fine. So we really just kind of created this opportunity to be as flexible as possible. And ultimately what this really led to is making me realize how we can take our science and do strong, rigorous science, but adapt to the needs of the child, whether that's doing testing at 5 a.m. And I don't always even see it anymore, but if you see in this picture, I am also wearing an EEG net because this child did not want to wear the hat, so I put one on and ran everything at the same time, and she did great. So, where are we now? So we've already, I think the count is 7 or 8. I think we might be running our 8th participant right now. And this is what it looks like. And some of you might be like, why are you doing EEGs in a hotel room? We've probably done 300 people in hotel rooms now, so I'm kind of super used to having my bedroom kind of be off to the side. But what you can see is we have our system that we put here in the corner. We kind of hid it behind the bench. We're running the computer here where you can kind of see the ongoing EEG. We have, this is Liv here who's helping kind of make sure hands aren't grabbing at the nets. And we have some very helpful parents who are able to entertain. I think that this was a Cocomelon special day. And it really takes a bunch of us, but we've done this very often, so we're very familiar with what your child might need. And the thing that we realized we really needed was Nico to help us with all the translations, and he has been fantastic. So if you see Nicola later, please thank him, tell him that I complimented him, because I cannot thank him enough for joining us. And just some more pictures of just how well we did. This is just yesterday. So if you haven't been convinced yet, let me know. I'll try to convince you afterwards about what it looks like. So what is it that we're doing during these sessions? There's a couple of things, so I'm just reminding you of kind of what we thought our targets would be, but we recognize the fact that our kids are not necessarily always going to watch the screen. So a lot of this other biomarker work that's being done in autism is trying to focus on things like how your brain understands a face versus a house. Our kids aren't necessarily going to look at the screen for 8 minutes, so we are really focused on things that are relatively quick, and that even if we get 2 or 3 minutes of data, that it's sufficient. So we have a standard battery, and we always try to get a resting state, which is the first thing that we start with, and I'll talk a little bit about some of those biomarkers. And then I'll also talk a little bit about our auditory attention task as well. We also have some really cool other adaptations that I could geek out about. One's looking at emotion regulation in a 3-minute test where we take a toy away and see how kids manage, and it's incredible so far. And we're also starting to do some hyperscanning where we bring in parents and child and siblings and child and look at 2 brains at once, but that's for another day. So let me start by talking through our resting state biomarker. And this is work that's being done by my graduate student, Sophie Kramer-Benjamin, who could not make it to Italy. So here we're taking the EEG, and we just heard amazing, I was taking furious notes about theta. So here, what I'm showing in this graph is this y-axis tells us how powerful the EEG signal is, and then as you go from like left to right, from like 1 to 40 here, you see the different frequency bands here. So when we were talking about delta earlier here, and I think the phrase was overshadowed by some beta, was it? So beta is here, right? So the idea of having a delta signal that maybe also have some beta on top. So when we think about this, what we can do is take different trajectories. So imagine that these two lines are different kids. And what actually we know is that these lower level frequencies are going to be more linked to inhibition, so GABAergic processes, and then these higher order spectral frequencies are related to excitation and glutamate. So if we think about this as that teeter-totter, that balance between excitatory and inhibitory activation in the brain, well, something that's a little bit less of a steep slope, this kind of more even seesaw, is what we expect to see in older kids, but we also see this in kids that have ADHD but are unmedicated, whereas these really, really steep slope are things that we see much earlier in development, but also for babies in particular that have more autism symptoms. So we have, and I should clarify, I realize it would have been super helpful for many of you to see our sample tables. I didn't think to prepare that. So we have, all of the data I'm going to show you is going to be the loss of function GRIN2B, but we've been collecting data and gain of function and unknown variants and variants of unknown significance too. So we also have kids where we're not entirely certain of the pathogenicity. So I'd be happy to talk about that data and show you some of our data afterwards. But for the most part, the data I'm going to present as just an example of the biomarkers is all loss of function. So here, Sophie worked with 41 of our GRIN2B data sets and compared it to autism without a known etiology and neurotypical development. And what she's finding here is, first, we were just hoping that the slopes would be different, but that rate of slope is actually not different at all between the groups that we did for this analysis. And what we did find is that the rate in which this slope changes with age is very similar across groups. So we actually see a conservation here. So I've kind of like marked this in like a yellowish finding. But what we did find, which I think is going to be related to this data that we just heard about too, is if you look at what we call the offset difference, so that's kind of thinking about where your starting point is, we actually see a much higher signal in our GRIN2B group. And granted, there is variability. You can see each of the kids plotted here. But this is telling us that there's this more neural activity. And now that I've seen this other data, you can see that we have higher delta signal, but you also see we have this higher beta and gamma. But really, when you look at alpha, which is where we spend a lot of my time in my lab, there's not much group difference. So I think that this is fantastic convergence across a data set. So there's clearly some markers here, I think, related to GRIN2B. So the other big category of biomarker that I've been chasing for about a decade now is thinking about auditory attention. So I wanted to just show you how we do that. And some of you parents who have already done this study, you will have recognized the fact that you heard these annoying beeps over and over and over and over again. So what you see is we have these frequent tones, and what happens is our brain gets used to this frequent sound, and then every now and again we have this pop-out effect, which is what we're calling a deviant here. And then sometimes you hear these like squelches and chirps and chimes, and that's a novel sound. And I don't think you can hear the sounds here, but this is one of our GRIN2B kids really enjoying listening to the weird noises. So we can look to see how much is your brain responding to these pop-out effects, but not only that, we also spend a lot of time thinking about how the signal changes across the experiment. And I won't talk about it today, but we have the same task also looking at speech sound discrimination, which is going to be a really nice link into language learning, speech perception as well. So what do? So we look at the EEG, instead of the whole EEG and the kind of spectra that I mentioned, we can say, what happens in that half a second after you hear a tone or sound, and look at the ERP and that time point. And what we see and focus on today in particular, and we can pull multiple things out. You can see a bottom hump here, that's important, but I'm gonna focus on this P3 response. And you can see that it's much bigger to this other condition, which is that pop-out effect. So what we can do in each kid is characterize what the condition difference is, and we predict that we're going to see this much larger amplitude to the novel sounds. And what we're seeing in GRIN2B is an actual, at the group level, a lack of that difference. So whereas in autism and neurotypical development, you see these really strong condition effects, and you can see it in a couple of GRIN2B kids, but at the group level, we're really not seeing this same pop-out attention effect as we kind of predicted. So what does this mean? Well, it's atypical in this way, but this is potentially consistent with that other finding I mentioned about the hippocampus. So there could be this learning piece where they're not necessarily attending to this pop-out in the same way. And because attention is so universal to all species, this is a really important potential avenue to really start to understand translational neuroscience. And I think I have enough time to talk through this, hopefully. Okay. So I want to talk a little bit about the work that we're doing to try to then bring it back into the animal to kind of really build that connection. So this is work that I've been doing with Dr. Annette Schenk, who's at Radboud Medical University and Nijmegen. Hopefully I didn't butcher that too much, thank you. So this is work being done by Laura Block. This is her dissertation. We've been working on this since 2018, so it's been a long time. And they let me sex flies at some point in time, so I feel like a little bit of a fruit fly expert now. So what we love is the fact that we kind of have this nice overlap where we're really looking at information processing and the physiology in both the human side and the animal side. So the first question we had is, can the fruit fly tell us what we expect to see in a large group of kids? So how the fruit fly behaves and responds cognitively, does that map on to what we see in our kids broadly? So they're using a Drosophila knockdown, and this is up. Do you mind playing the video? This is just Laura. She puts each little fly into one of these chambers. And then we turn all the lights out, and this is a startle response. So it's kind of light on, light off. It doesn't necessarily matter. And what we're looking at here is every time the lights flash, how startled are the flies? And you can kind of see that they start by jumping all the time. Oh, thank you. And then it kind of slowly habituates. And you can see this top line here is the control, and these are just the smooth graphs. And this bottom line is one of the mutant lines. So what we're trying to understand is how does this differ in different groups. So we had a collection of different flies that responded in what we're calling a severe way, where they actually habituated way too soon and were way less likely to, they took fewer trials to habituate to the stimuli. And you can see some of those genes here. We only saw one gene where the mutant fly performed the same as the wild type, and that was the Kismet or the CHD8, CHD7 gene. And here is where we find our GRIN2B in what we call this atypical pattern. So this is actually very similar to things that we've described in autism, where we're maybe seeing what I like to call sticky attention. So these flies almost, like they do kind of habituate, but still a lot of them are responding across time. And you can see here that the trials to habituation is still quite heterogeneous, and this is for the Direxin fly, but this is the same pattern that we're seeing in GRIN2B. So thinking about how this maps on, what we can do is we can take that P3A response, and what you see here is two different children. These are from an earlier paper of mine, and you can see I have the kind of overall what that child's ERP looks like in red here as that thick line, and that's usually where cognitive neuroscientists stop. They say, here's this person's response. But we take every single time they hear a sound and are taking that and plotting it over time to look to see how does that attention signal modulate in time as a proxy for learning. And when we look at the group that had this fly atypical pattern, including the GRIN2B, we're seeing this weak attention effects and habituation. So there is attention differences early on in the experiment, but you see as you get closer to the end of the experiment, here at 100% done, we're seeing less difference. So this is kind of showcasing the way that that pop-out effect is changing. Oh, I forgot I animated this little fly. It's very different to these other groups. So I want to dig more into the GRIN2B. So we know that we can use the fly in this way, which is really exciting, but then we really wanted to understand that specificity. So how well is this going to align specifically for GRIN2B? And so here's just that gene group plotted, and what you see is really a pretty flat slope. And I can tell you that when you do the statistics on it, there really is no change at that group level. But we're seeing this kind of alignment. So while we saw kind of a lack of habituation, kind of a stable response across the experiment, the GRIN2B flies were also slower to habituate. So we're seeing this as convergence for both for the GRIN2B group in particular. But then I really, because I want this to be a biomarker, so I need it to matter for each child. So I thought, let's look at the individual data. And this is something you're warned not to do very often as a scientist. And I'm warning you now that it's a little bit mixed. But here are all of, at least the first set of kids. And that looks super wild to you, I'm sure. So what I want to do is just highlight that these are the individuals that are showing that same fly phenotype, this slower to habituate, lack of habituation. So it's actually about half of the sample was showing this lack of habituation, whereas you can see a couple other cases look a little different, where you're seeing some early habituation here, and I see this as sensitization, where you're actually becoming more interested. So we clearly have a little bit more work to do to really understand this, but the fact that there is at least a majority of the sample looking like this is a nice proof of concept. So where are we? So we are really kind of trying to understand, like, what is the functional reason for this. We're talking about one process. We actually have the capacity with 128 different channels to understand more of what's happening inside the brain. So that's work that we're doing to try to understand kind of what neural mechanisms or what brain regions are involved in these pieces. And then we're in the process of linking it to different clinical features, as well as trying to understand other biological factors, including sex, like we had the conversation earlier. And then my big question is, how developmentally stable is this? Is this something that shifts during puberty, for example? So we have a little bit to uncover, but what we really hope is to say, okay, so if we can use habituation as a learning metric, and we can link it back to the flies, where we can do these really quick drug assays, and we see this one-to-one alignment in my little toy example. Well, we had that severe group, and we had our atypical group with GRIN2B, where we're seeing this kind of increased sticky attention. Well, we know that there's different interventions for each. So if we're measuring the fly, we could still look at their habituation and look to see how those drug interventions work. If we're working with our kids, we can look to see, well, first of all, how is the drug adopting in them? But we can also look to see things of other more common therapeutics. So is ABA actually helping them manage their environment and able to attend in a more important way? And really thinking through what the optimal recommendations are for treatment. And then I just wanted to end with the things that are always in my brain, and these are actually very similar to a slide I presented 2 years ago, so we still haven't solved very much, but I just want to emphasize the fact that we are really excited to expand into other GRIN-related and GRIA related disorders. I think that working together and maybe thinking about how we can combine our data sets is going to be absolutely essential. It's not necessarily the easiest, but I think it's really doable, and we've got some energy, so let's try for that. So I just want to thank everyone for participating. I'm going to leave this little QR code up for one last second. And again, please make sure to thank Nicolo and Liv if you see them. They really are essential to the work that we do, and I thank you all for your attention.